Hi there, fellow primates. I hope this video finds you well. Recording this on Wednesday afternoon, 15th of December. The light is already fading around 3 o'clock in the afternoon. I better be quick because otherwise the picture gets too grainy. Now, got my booster shot yesterday. I got a shot of Pfizer in my left shoulder. Bit of a sore arm, but I feel fine. I cycled to the museum this morning and back, a 28-kilometer round trip. Much better than the AstraZeneca shots I got before. The first shot really knocked me out for 2 days. The first day I was in bed shaking with fever and muscle pains. It was horrific. So this is much better and well organized, because there was a lot of stuff in the media about people having to queue for hours in walk-in centers. And maybe it's because I went to a GP surgery that was not a walk-in center. I was helped within 5 minutes of walking in the door, and I was allowed to go 15 minutes later after they observed me, and it turns out that nothing was wrong with me. So excellent, well done, Croydon. Today I want to talk about my rules for writing. This is a follow-up to the video that I released earlier, and it features some of my biggest peeves, my biggest annoyances when I read scientific papers. So the rules here are numbered consecutively following up on the first video, and the first one is, or number 10 is, don't make everything about us. What do you mean, Ronald? Well, for instance, take this 2014 paper in Science, written by 106 bird-loving authors. It's a real benchmark. It was the first real genome-level phylogenetic study of birds, and it analyzed the evolutionary relationships between major living lineages of birds and the time in evolution when they diverged. Beautiful benchmark work has been cited now more than 1,000 times. And as you can see here, this phylogenetic tree is one of the major findings. It gives a real deep insight into bird evolution. Now, then you start reading, and you get to the last sentence of the first paragraph of the introduction, and then you read this. Resolving such timing and phylogenetic relationships is important for comparative genomics, which can inform about human traits and diseases. What? Well, actually, maybe it's good, because we should perhaps justify all this weird intellectual work with respect to ourselves. It's perhaps good that we benchmark everything we do against the only universal standard, namely us. So maybe that's great, right? So then you start reading on, and you come to the section where they justify what species they sampled. And you read this. Well, we included vocal learning species such as songbirds and hummingbirds and parrots. They are used as models for spoken language in humans. Really? Really? Jeez. And this is really, really annoying. Why? Well, of course, when we try to get grant money in this highly competitive funding world, yes, we try to relate the research often that we do to human benefits, economic benefits, societal benefits, medical benefits, blah, blah, blah. And that is justifiable up to a point. But then once you got the money, you got the research done, and you got it written up, and it is good enough to submit to the journal Science, to then benchmark what you're doing in the introduction and the materials and methods against humans. You are doing a large phylogenomic analysis of birds. It's complete madness. At best, this is silly. At worst, it's pathetic. It is just childish narcissism. Why do so many researchers continue to use Mother Nature only as a distorted mirror in which we can desperately try to see our own reflection? Stop doing that. Study Mother Nature for her own sake. Second, or perhaps the 11th rule, present raw stats in tables and charts. What do I mean with this? Well, check this paper out. It is titled In Purgé, Sexually Dimorphic Venom Proteins in Long-Jawed Orb-Weaving Spiders Comprise Novel Gene Families. Really cool paper by Pamela Zobel-Throp and Greta Binford, colleagues whom I've met. But then you start reading their results. First paragraph of the result reads like this. The final trimmed read files comprised 43,550,457 females, 20,370,960 left, 20,370,960 right, 2,808,555 merged, 48 million... Yes, I'm not going to continue this. Put this stuff into a table or park it in the supplementary materials. That's what these things are for. Don't put raw quantitative stuff like this in a narrative sentence. In fact, when you write your paper, think that you're writing it for humans, and this doesn't read well. It's not presented in an ideal format. Try to remove elements from your narrative that are not easy on the eye as narrative element. It just makes the readers happy if you do that. 12th rule, only present informative data in figures. Well, surely, that's what people do, right? Researchers will put all the images in that they think are informative to support their research. We do, by and large, often. I think that scientists are actually really good in visualizing, because don't forget we are a visual species, results that are often purely quantitative. We do that well, but often we don't do it so well. Take this paper. It's on ant venom. And then look at one of their figures. They try to compare venom composition between ants that live in trees and ants that live on the ground. You can see these little overlapping circles. And then with their mass spectrometry techniques, try that one more time. Is it spectrometry? Yes, spectrometry techniques. They look at the molecule masses of the things that are in common in the venoms of the tree-dwelling and the ground-dwelling species. And there they are. An enormous list of 377 long numbers. Well, that is a waste of space, isn't it? Because no one who reads this information will actually retain it because, or get anything out of it. Moreover, researchers who are interested in molecular masses like this, they want to do follow-up research, they would like to have this presented perhaps in a usable format, a table perhaps, an Excel spreadsheet, which they can use to manipulate the data and get it. This reduces researchers who are interested in this to having to figure it out by reading it, because it is embedded in a picture file. That's not very good. That's a wasted opportunity. On the one hand, it is overkill, presenting way too much information for a human to absorb. On the other hand, it is completely useless because it's offered to you in the wrong format. Naughty guys, don't do it. I've got another example, and this is about snake venom, about the venom of the Malaysian king cobra. And check this figure out. This looks at the transcriptome, all the expressed genes in the venom gland, and then you can see two pie charts, one of all the expressed genes and the second larger one of those expressed genes, all the transcripts that code for putative toxins, venom components. And then you look at it, it's like, well, I'm quite interested in NGF. I think that's nerve growth factor. But I'm also quite interested in hyaluronidase. So how much? Well, they have no numbers on this. And I'm not even colorblind. This is not very informative. Everything that is not purple in a large pie chart, you can't figure it out. In fact, some of them are probably such a small proportion of the genes expressed in the venom glands that you can barely see them. And you would have to browse through the article to a different table, which is filled with other information, to get the actual numbers. Guys, put the numbers in so you can actually make this image informative. It ain't informative now, it sucks. But it can be worse. If the first picture I showed was overkill in a way, and then this is underkill, I looked it up, that's an actual word, then this one is actually deaf, blind, and mute. What do I mean? Well, this is a paper on spider venoms, and they analyzed the different spiders and want to see if the venom composition has any correspondences. And they produced this phylogeny, the evolutionary relationships of venom components in these spiders. I don't know what this is. Well, in the legend it says the red branches are the one species. Oh, great. And the black ones are the other one. That's great. Oh, there's 9 clades, are they? Yeah. And then if you look at the names at the tips of the branches, these are just codes. XP4457Q. I don't know what that is. And if you read the article, neither do they. So it is unclear what these clades are, or what kind of putative homologies there are. You can't get anything from this figure. A test that you should do if you want to include an image in a paper is to think, if I see this paper and I'm sitting in a room with a colleague and that colleague cannot see the paper, can I describe what the image shows? And the 3 images that I showed, you can't. Well, you can say here these 2 spiders, they have toxins and they have clades of things that we don't know are homologous and even belong in the tree. But did I say that some of the branches are red? Yeah, no, that is a large figure, more than 1/3 of a page, that could be put in the bin, or, you can make this informative, but I'm not going to try to explain that to you. This is a bit shit. Then we get to rule number 13. When you have images, and you have good images, you can let them speak for themselves. And this means that, we all labor when we write papers under the restrictions of word limits. You want to reduce redundancy between text and between images. So if you have a good image, the reason you have the good image is that you can save yourself typing a lot of stuff. So make a good image, put a good legend in there, and then you can reduce redundancy. But often, especially in the papers that are molecular phylogenetics, but also transcriptome studies of venom glands, in which people look at the genes expressed in venom glands, you often read the text, and what you read is a description of what is presented in figures. The pie charts or bar charts show stuff very clearly in a visually appealing and intuitively easy to understand way, and then there's long narrative paragraphs describing mostly what's already in the figures. Don't do that. Nobody wants to read that stuff. It makes us very, very bored. And the same with clades in molecular phylogenetic trees. Some authors feel that they need to explain in words what the trees show. You don't. And to remember this particular point, my favorite image of something that doesn't need words and in fact speaks louder than 1,000 words comes from a paper in the Journal of Forensic Science. It's titled Centipede Inflicted Postmortem Injury. And they found in an apartment a dead Japanese man, and it had been scavenged upon a little bit by a large centipede. And if you check this image, it says figure 1, the bedroom where the corpse to the right of the picture was found. Now we can see that is to the right of the picture. You don't need to put these words in. We can see that the corpse is there. We don't need more precision than that. And this particular picture shows quite a large centipede, actually. This one, it lies there very still because as the police came to the apartment to figure out what was going on, a centipede came out of a small hole in the dead guy, and a police officer went and stamped on it. And so this is the treaded upon scolopendra moth. It's dead. Brings us to rule number 14. Show a picture of your organism. Oftentimes, when people work on model organisms, you don't need to show a picture of a zebrafish or a mouse or a nematode because most biologists know exactly what they look like, and there will be an avalanche of pictures you can get online with the click of a button about these creatures. But many of us who work on non-model organisms, well, if you write about your organism, don't let the curse of knowledge afflict you. By which I mean, don't assume that your readers know what your creatures look like. Show them, because first and foremost, we like organisms. That's why we are biologists. So when you look at this nice paper by Marco Cerdal et al. On a vampire snail, I was like, oh, what is a vampire snail? And there's no picture of a vampire snail in this paper. It's just graphs and gene sequences, which are important, but I would quite like to see a vampire snail. Does it have fangs? Does it have a wee cape? Now, a paper who did do it very well, and I should hide this, otherwise I can see it on my screen, is by Dominique Coué et al. A paper on venom in a wolf spider. And they have this beautiful image of the actual spider, of its burrow, and then a schematic of the morphology of the venom system. Excellent. This is very informative, and that should almost be mandatory, I think. Now, the way I have organized on my screens all the little images to remind myself the sequence of things I want to talk about, they're now underneath my camera. So I'm going to try to hide them, the camera that is, so I can actually see what I'm doing. Rule number 15, cite relevant literature, and that is very important. It's also difficult to navigate problems sometimes because many journals don't only impose a word limit, they also don't want you to cite too many papers, which in these days of open access and online publishing is an argument less and less convincing. But if you then look, for instance, at a paper like this about centipede venoms, topic I work on, so the next examples will be about centipedes. It says in the abstract, very proudly, we are the first group to report the gene recruitment activity of venom toxin-like proteins in centipede, similar to snakes. Not fantastic English, but I understand what they are saying. They claim to have found for the first time examples of toxin gene recruitments from housekeeping genes in centipedes. Yeah, that's absolutely true. I mean, I congratulate them on it. Except it isn't true if you actually read the literature. This is a paper published in 2018. They ignore the work by a PhD student from Australia at the time, Ivan Undheim, a colleague of mine and a friend of mine, who did a lot of this work years before. They don't cite that stuff. They are not the first at all. In fact, they ignore a whole host of unique work done years before them. That is a bit unscholarly. I mean, you can't really say, oh, well, I didn't know it existed. You go on the Web of Science, you type in centipede venom, you can't miss it. So I don't know what the rationale here was for not referring to pre-existing literature. It actually pissed me off so much I wrote to the editors of the journal, but of course you get no reply. Honor the work that your colleagues do inside their work, please. 16. Do your effing homework. What do I mean by that? Stay with centipede venom. This paper in the Journal of Peptide Science, write this in the introduction, trying to tell the readers what creatures they work on. And they say this, they say centipedes are predatory terrestrial arthropods that feed mainly on vertebrates, including reptiles, amphibians, rats, bats, and other insects. First inference, these are not taxonomists. Second inference, they didn't do their homework. Centipedes are not primarily terrestrial arthropods that feeds mainly on vertebrates. Most centipedes, even the large ones, feed on invertebrates. There's maybe only some very specialized, very large-bodied species that put a good proportion of vertebrates on their menu. The rest don't. This is just a overgeneralization at best. And we come to the last rule that I want to discuss today. It's an important one, and I can't phrase it in a friendlier way than this. Don't bullshit your readers. Surely bullshit will be filtered out by peer review. I don't think so. Check out this paper, 2018, Proceedings of the National Academy of Sciences of the USA. Big high-impact paper, and it's about how centipedes can subdue giant prey by blocking certain ion channels. Excellent. And then, well, that's very interesting. So you read the paper, and they constantly talk about these giant prey, which are mice, bigger mice than the centipede, and how strong is this centipede venom? Until you go to the snuff movie that is included in the supplementary information. And then you can see the relative sizes of the mice used in the experiments and the centipedes, like this one. Or this one. Now, what I'm impressed by is that there's something giant here, but that's not the mouse. It's the effing centipede. It's an enormous centipede. And then you read the legend of the movie, and it says a single golden-head centipede weighing 3 grams attacks a mouse 45 grams and rapidly subdues it. Bullshit. Unless this centipede is hollow, there's no way that this centipede, which is as long or perhaps even a bit longer than a mouse, weighs only 1/15 of its weight. 3 grams is not even a full teaspoon of sugar. This is bollocks. This is just sensationalism in the interest of advocating rather spectacular findings. I mean, check the snuff movie out. It is pretty horrific to watch. And the centipedes can subdue prey. Perhaps a small one can subdue a mouse 15 times its size. But that paper doesn't show it. And that brings me to the end. And that brings me to a saying by this guy here. This is the late Christopher Hitchens, journalist, British-born, naturalized in America. An unbelievably good prose stylist and writer. And he wrote in a different context, in a book titled God is Not Great, something that is perhaps relevant here. And he wrote, what can be asserted without evidence can also be dismissed without evidence. So if you try to BS your readers, then you run the risk of them thinking this about you. Now, why do I bring up Christopher Hitchens today? This is why I'm recording this on the Wednesday. Is the 10th anniversary already of his death. He died of cancer in 2011. And I want to close with 2 short things I want to read out. Why? Because for me, as a non-fiction prose writer, as many of you viewers may be, if you're a scientist and you write papers, and some of you may write books, he was a great example of how to construct beautiful, powerful sentences. He was a great essayist, a great polemicist, very good public speaker, and probably one of the scariest public debaters I've ever seen. You would never want to be on the other side of this guy. So I want to read to you 2 little things that explain his passion for his craft and the motivation to do what he did right until the end, shortly before he died. So the first thing I want to read to you is a paragraph from a book called Letters to a Young Contrarian. which is an imagined correspondence between Christopher Hitchens himself and an imagined younger colleague who wants to become a critical writer himself. And it reads, "How to ward off atrophy and routine, you ask? Well, I can give you a small and perhaps ridiculous example. Every day, the New York Times carries a motto in a box on its front page: all the news that's fit to print. It says, it's been saying it for decades, day in and day out. I imagine that most readers of the canonical sheet have long ceased to notice this bannered and flaunted symbol of its mental furniture. I myself check every day to make sure that the bright, smug, pompous, idiotic claim is still there. Then I check to make sure that it still irritates me. If I can still exclaim under my breath, why do they insult me? And what do they take me for? And what the hell is it supposed to mean unless it's as obviously complacent and conceited and censorious as it seems to be, then at least I know that I still have a pulse. You may wish to choose a more rigorous mental workout, but I credit this daily infusion of annoyance with extending my lifespan." Not that much though. He died sadly only at the tender age of 62. So that's a very sad anniversary that we are celebrating. I wonder what happens if there is a God, and if he manages to get there when he compared heaven to a celestial North Korea. Some conversations that must produce up there or down there, wherever heaven is. But I want to close with something that his friend Ian McEwan, the great British novelist, wrote. He visited Hitchens in the hospital in Houston shortly before his death. And Hitchens' son Alexander was there too. And this was a piece from the New York Times. And I just want to read this because it illustrates what passion looks like in a writer. The next morning in the hospital, at Christopher's request, Alexander and I set up a desk for him under a window. We helped him and his pole with its feed lines across the room, arranged pillows on his chair, adjusted the height of his laptop. Talking and dozing were all very well. But Christopher had only a few days to produce 3,000 words on Ian Kerr's biography of Chesterton. Whenever people talk of Christopher journalism, I always think of this moment. Consider the mix: constant pain, weak as a kitten, morphine dragging him down, then the tangle of Reformation, theology and politics, Chesterton's romantic, imagined England, suffused with the kind of Catholicism that mediated his brush with fascism and his taste for paradox, which Christopher wanted to debunk. At intervals, Christopher's head would droop, his eyes close. Then with superhuman effort he would drag himself awake to type another line. His long memory served him well, for he didn't have the usual books on hand for this kind of thing. When it's available, read the review, and it is included in an essay collection of Hitchens called And Yet, which you can get on Amazon and elsewhere. His unworldly fluency never deserted him. His commitment was passionate, and he never deserted his trade. He was the consummate writer, the brilliant friend. In Walter Pater's famous phrase, he burned with this heart gem-like flame, right to the end. So with that, I want to close today's video. It's more than long enough. And as you saw in the picture, and I will put it up here again, of Christopher, one of the fuels that he ran on was Johnny Walker Black. I have a pre-prepared portion for me as well. Thanks for all your writings, Christopher. I don't know if you're up or down or nowhere, as I believe, but thanks for your inspiration. Your words are still there, even though you are bodily not. Thank you for watching. If you like this, like it, subscribe, sign up for alerts, and thanks for watching. See you next time.